All right, y'all, continuing on with our conversation on our second lines of defense. We've so far talked about interferons, the process of phagocytosis, the process of inflammation, and the process of inducing a fever. So now we're going to move on to a few more components of our second line of defense, and these will be a little bit less involved, I think, than some of the ones in the previous videos. So first up, natural killer cells, which we normally just refer to as NK cells. And these are where that idea of differentiating self cells from non-self cells really comes into play. So how do they do this? Well, it turns out that there's a certain molecule called MHC1 that is on the surface of every single self cell. And so what you're seeing in this top picture is an NK cell has stopped a self cell, and notice it can tell that this cell belongs because it has an MHC1. Okay, and so notice this is called the inhibitory receptor because this binding here is going to inhibit an immune response. We do not want our immune system to respond to our own cells, right? So when this inhibitory receptor comes into contact with an MHC1, that tells the NK cell, hey, this cell is good, it belongs here, let it go. And it does, okay? But what's important to know is that bacterial pathogens, viral pathogens, they don't have MHC1, but it also turns out that infected cells, like you see here, when cells are sick, whether it be because they're infected with a virus, maybe there's cancer, they're cancerous cells, it turns out that those cells have a decreased number of MHC1. Sometimes they don't have any at all, okay? And so when an NK cell comes into contact with either a foreign cell or an infected self cell, notice there's no MHC1s for it to interact with. So in that case, it interacts with something else on the surface of the cell, and it's going to activate an immune response. So notice here, the cell will die. Now, I want to please make a note of this. This makes it look like the cell blows up, which is misleading because it does not. Infected cells, when they're targeted for destruction, we introduced this in the last video called apoptosis, the cell is going to implode. That way, if it is viral, like a virus is inside of it, it won't be able to escape and infect other cells, okay? So when apoptosis occurs, the cell implodes. It does not explode, okay? So how does the NK cell cause the infected cell or the pathogen to die? Well, the NK cell has a couple of different weapons of its own. One is called a perforin, which you see here on this third bullet. Perforins literally like punch a hole in the membrane of the infected cell, or if it's a bacterial cell, whatever the pathogen may be, but it literally punches holes into that target, which allow little proteases to get in. Remember, proteases, we know they're enzymes. Prot lets you know that they target proteins. And what these little proteases do is they go in and they, destroy certain proteins, which leads to the process of apoptosis, which again is where the cell collapses or implodes and it dies, okay? So notice naturally killer cells do target bacterial cells or like host cells that are infected by viruses. They can also detect cancerous cells. And so that's what you see here. Natural killer cells are attacking a infected host cell. Kind of cool image, kind of creepy all at the same time. All right, next up are neutrophils. Neutrophils are another type of white blood cell or immune cell. And these release what are called nets because they act like a big old fishing net. So NET actually stands for Neutrophil Extracellular Trap. So what extracellular means? Extra means outside of the cell. So before I tell you exactly what happens, take a look at this picture. Right here where my mouse is, that's the neutrophil. Okay, that's the cell we're focusing on. And over here, these redishy orange, these are little bacillus bacteria. So this is the target, this is the pathogen. Well, do you see how with our neutrophil here, do you see this kind of green net web-like stuff that's shooting out and covering the pathogen, the bacteria? That's the net. So this is something that the neutrophil makes and throws outside of the host, outside of the cell, okay? And it's acting like a big old fishing net. It's trying to cover up and tangle up these bacterial cells. That way they can be killed, okay? But here's the catch. These nets are actually made up of this neutrophil's DNA. Okay, so the neutrophil does not survive once it releases this net. It's essentially sacrificing itself. Okay, so what happens is once the bacteria is detected, this neutrophil is able to find it, and then it takes its DNA and it releases it out. It captures it, like you see here, and then these really aggressive enzymes follow suit. They can use the net to get to the bacteria and break them down. Okay, so it traps and eliminates that pathogen. So this is a very common target for extracellular pathogens, things like bacteria and fungi that stay outside of specific host cells. Okay, so nets made up of DNA and aggressive enzymes that are released from a neutrophil to trap and kill pathogens. All right, next up is going to be the complement system. Notice, y'all, it's called a system. That's because there are actually 3 different pathways made up of over 30 different proteins that work together to detect and destroy pathogens. Typically, they are viruses and bacteria, but the outcome of the complement system can work on other pathogens as well. So this picture you certainly do not need to memorize, but notice there are 3 different pathways, the classical, the lectin, and the alternative. And each of these has a different target, a different thing that it's looking for, okay? So with the classical pathway, this is when an antigen and antibody find one another. And this will make sense more after we discuss specific immune responses. The lectin pathway is looking for sugars that are only found on pathogenic surfaces. So this pathway gets activated when our body detects sugars that shouldn't be there. And then the alternative pathway just detects other things on the surface of a pathogen. So whichever of these gets activated is going to cause a cascade reaction, which is where, like you will see here, you know, C3 protein is going to get broken down into C3A and C3B, which then reacts with C5, which reacts with C6, C7, C8. Okay, so again, you don't need to memorize this, but it's called a cascade reaction because one protein goes and activates another protein, which activates another protein, and it's kind of like a domino effect, okay? But what you'll notice is no matter where, or I guess which pathway gets activated, because notice up here at the classical pathway, we're starting with one protein. The lectin comes in at a different point in this cascade. Alternative comes in at a different point. But no matter what, all 3 of these pathways have the same 3 consequences: opsonization, inflammation, and cytolysis. Okay, so that's what's down here at the bottom of the PowerPoint. All 3 pathways, doesn't matter which one gets activated, all of them have these same consequences. So opsonization is a new word. We've heard of inflammation, so I'm not going to elaborate on that one in this video. But opsonization basically means the pathogen gets covered with protein, okay? And so you might be thinking, why do we want to do that? Why are we covering a pathogen with protein? Because that helps enhance phagocytosis. Remember, I told you a lot of bacterial pathogens have a capsule, that sugary, sticky structure that can make it hard for phagocytes to latch on and catch the pathogen. Well, we fight back against that capsule by covering it with protein. So look at this little rectangle here. So this little green circle is a complement protein. The little gray oval is our little bacteria. Let's pretend it has a capsule, a sugary structure. Well, we need our phagocytic cells to be able to grab hold of it. So we coat it, we cover it with this complement protein. These are going to serve as like little handlebars that the phagocyte can grab hold of so it can capture this pathogen, and then phagocytosis can occur. Okay, so we're going to coat these pathogens with protein. The third outcome is cytolysis, which means it is going to essentially blow up the pathogen. And this happens by forming a membrane attack complex. That's what MAC is, membrane attack complex. And so this is what you're seeing here. See how there's the letter C in front of these numbers? The C stands for complement. So this is complement protein 6, complement protein 7, 8, and lots of 9s. Okay, so all of these complement proteins that you see here work together to form a pore in the pathogen membrane. Okay, so this is a bacteria. These proteins stick themselves into the membrane, forming a pore. That way, the cell becomes leaky and eventually it lyses or explodes. Okay, so that is our complement system. Last but not least are going to be antimicrobial peptides. So when you see peptide, you should be thinking peptide bond, which are the bonds that hold proteins together. Okay, but these are really teeny tiny. I'm not even going to call them a protein. They do have a 3D shape, but most proteins are about 500 amino acids long or more. Notice these are 12 to 50 amino acids, okay? So these are very, very short, but they are proteins, so I guess I can ease up on that. So short proteins that are amphipathic, because look here at what they do, okay? So we're looking at a bacterial pathogen's membrane. This purple thing here is the amphipathic antimicrobial peptide. So remember, amphipathic means it has hydrophobic and hydrophilic regions. So here we can see these are the hydrophilic portions of the antimicrobial peptide because it's interacting with the hydrophilic head groups of the membrane. Okay, but look at this region down here. This is hydrophobic because it's inserted itself in between the hydrophobic portion of the membrane. So because it's amphipathic, it will be attracted to the membrane and then be able to wiggle part of itself in between it, again, causing it to be leaky and allowing the bacterial cell to explode. Okay? All right, that is the end of our non-specific immune responses. Please let me know if you have any questions.